Yesterday at 1 p.m. was a really exciting day because it was going to be when I defended my thesis after many, many years of graduate school. And then what ended up happening was, as anybody here with a Ph.D. knows, trying to get 4 professors, your committee, in one room together for 2 hours is harder than the defense itself. So a week ago, I got an email saying, I have a really important meeting. I'm really sorry. Can we push your defense back? So I just had to begrudgingly say yes. So July 22nd now is the official date. So, so I was going to make the PhD letters pop right in there, right, as a joke before the talk. And now I can't do that, so maybe next year. So I'm actually amazed at the resonance between the last talk and the kind of stuff that I'm about to talk about in mice. The kind of work that I've been doing at MIT focuses on trying to find individual memories in the rodent brain and then trying to tinker with those memories. Can we turn them on or off? Can we get them to do what we want them to do? And recently, could we actually apply this to potentially have some therapeutic value? So we're going to quickly start off just a brief overview of the kinds of memories that I'll be talking about, and then I'll specifically talk about a recent project that we've had on using, I think of it as sort of weaponizing memory and actually using it as a weapon against certain symptoms associated with psychiatric disorders. So a nice little twist on treatment. Now, why do that? Why is that something that I personally considered important? We've known for a long time, and nicely written through commentaries from Steve Hyman and Tom and so on, that the 1950s and 60s, very real revolution. Psychopharmaceuticals, the works, all of our drugs that exist today are basically based on iterations that were discovered in the 50s and 60s. That's good and bad. We need the drug research. We have to know what they're doing. We have to know how to improve on them and so on. The problem is when you get too many copycat drugs, and now you basically start almost becoming a little bit dogmatic about particular interventions, and we stop asking, what else can we do? What else is there? What other kinds of information, what other kinds of novel interventions can we produce to try to get the brain to actually become fixed? So what can we do? Since I worked in a memory lab, one of the things that we considered was, well, what if we, rather than using a drug, what if we go and actually try to go within the brain itself and harness the endogenous processes that can occur in the brain and force the brain to fix itself? How do we do that? Well, for starters, we can use things like positive memories. So positive memories are something that we inherently find them rewarding. For me, like the Red Sox winning the World Series, wildly rewarding. Trip to Cape Cod. I was going to have a Patriots thing up here, but if anybody mentioned Deflategate, I was going to get mad, so I figured I would just leave that out. And so they're inherently rewarding memories, and the question that we have was, okay, now can we go in, can we get leverage over a particular memory, and a positive memory, and turn it on? Can we find the brain cells that we think are holding on to a component of that positive memory, and can we trick those brain cells to turn on? So to briefly go over the system that we use, so this is all combining optogenetics and transgenic animals, but to briefly go over it, the way we identify cells that are active during a specific mnemonic task, for example, is we couple the promoter of the immediate early gene, the activity-dependent gene CFOS. For all intents and purposes, when a brain cell is active, CFOS is active. We have it drive TTA, tetracycline transcriptional activator. So TTA binds to that which it binds to, which is the response element, the tetracycline response element, TRE, which ultimately upregulates channel rhodopsin, which is our nice little light-sensitive switch that we can artificially install in brain cells. So complicated system, but the whole point of this is that it's regulatable by the drug doxycycline. If we raise an animal on a dox diet, none of these steps happen. No cell that's active during a mnemonic task contains channel rhodopsin. We remove dox from the diet, this entire cascade of events can happen, and cells that are active during a particular memory task, for example, upregulate channel rhodopsin. So that way we can open and close windows of when we label a particular set of cells in the hippocampus, for example, and multiple other brain areas. So, okay, so that's the little genetic trick that we use to find brain cells active during a defined period of time and trick those brain cells to respond to pulses of light. My favorite picture I've taken in all of graduate school was actually my first year. I consider it a cross-section of what a memory looks like at the level of the brain. For various reasons, we're working in a subregion of the hippocampus, the dentate gyrus, and this is what it looks like. I think whatever the realization of a memory is at the biological level, this is a nice cross-section of it at the level of the hippocampus. Okay, so To quickly update everybody on where we have been and where we are going, and to convince you that this is an aspect of a memory that we're reactivating, it started in 2012 where we were able to show that cells that were active during fear learning, for example, were sufficient to drive that fear behavior when they were optogenetically reactivated. We later went on to show that we can change the contents of a particular memory, turning a neutral memory to a fear memory, for example, or, very importantly, that it's not just specific to fear, but that we could also activate reward memories, positive memories, what I'll call. Very interesting project that's recently come out from another collaborating dynamic trio in our lab showed that we can bring memories back from a state of amnesia. And then the project that I'll talk about today was just published last week, and it's now asking, can we use these artificially activated positive memories to suppress a medley of symptoms associated with depression to the extent that we can model that in animals? So, okay. A positive experience for a mouse can be many things. We chose nature's most natural social reward, which was exposing male mice to female mice. They go bananas over it. They love it. If you've been to a bar at 1 in the morning, you know exactly what that behavior looks like. You can tell what's happening. So we go in, we expose the male mice to the female mice, we label the cells in the hippocampus that are active during this positive or rewarding experience, and we trick those brain cells to respond to pulses of light. So another dynamic duo in the lab showed that we can induce artificial place preference by reactivating those cells in a component of a box, as measured by difference scores. That's just to convince you that we can go in and activate cells that were previously active during the encoding of a positive experience. Again, that's the weapon that we needed to make this last project work. So that's the hypothesis, that hippocampal cells that we think are encoding an aspect of a positive memory are sufficient not just for the reinstatement of that positive memory, but to rescue maladaptive behavior, specifically stress-induced maladaptive behavior, since that's generally one of the most well-characterized methods of precipitating certain depression and anxiety-like behavior in animals. So the tests that I'll talk about, with the 1,001 caveats that these tests actually have, one was just measuring motivational levels or active versus passive action patterns in a challenging situation. So the tail suspension test, primarily used as a screen for antidepressant treatment, right? Not a measure of antidepressant behavior. And the inability to seek out pleasure or anhedonia, one of the core symptoms of depression, and of course a variety of anxiety-like behaviors. So moving forward, does this work? And if so, why? The caveat here is that, of course, mouse brains are not human brains. Where we have wrinkly brains, they don't. Ours are bigger, theirs are smaller. There's a lot of things that we know we can't model in animals: guilt, sadness, suicidality. There are certain things that I would argue we can model at a rudimentary level, certain behaviors, motivation, anhedonic-like states, and so on. Okay, so there's a lot of groups, and there's only 3 groups I'd like everybody to focus on. There's a group where we label this positive experience, or a neutral experience, or a negative experience. That's important, right? Because valence can matter. And then we stress animals out by giving a chronic immobilization protocol. That precipitates a medley of these depression-like and anxiety-like behaviors. And then we put them through a battery, and then we shine light and reactivate these cells and ask, what are the behavioral consequences? So the first group here, and the most important one, is the orange group, showing first that stressing these animals out decreases time struggling during the tail suspension test. And then as soon as we turn on the laser and we shoot light into the brain to reactivate these hippocampal cells previously associated with a positive experience, they dramatically increase struggling back to the level of control on stressed animals. And importantly, reactivating a neutral memory has, it almost has an effect. It's very mild. And reactivating a negative memory doesn't seem, I mean, it might be doing something, it might not be doing something. This could be a floor effect. But the point is that valence matters, because this doesn't rescue the effects the way a positive memory does. So, here's a video, see what it looks like. So this is a stressed animal. You can see that they're not really that motivated to escape. There's some basal levels of struggling. And then as soon as we turn the light on, you'll see instantaneously the animal shows this escape-related behavior. I would show the sucrose preference task also, but it's a 30-minute task, and I have 20 minutes to talk, so it wouldn't make sense to show it. Okay, so for the sucrose preference task now, as a measure of hedonic-like activity, right? Normally, unstressed animals would choose sugar water 80% of the times whereas animals that go through this chronic stress protocol show a 50-50 preference for sugar water and regular water. So we look at that behavior and say that it's mimicking the inability to seek out naturally pleasurable stimuli in the world. So this is what the task actually looks like. There's sugar water, regular water. Here's our animal plugged into the laser. The problem with this task is that our particular strain of animals simply wouldn't lick these lickometers. So we spent 9 months trying to troubleshoot this task, trying to get our animals to lick. There was 1,001 iterations that we tried. A baby could have happened in that time. And anybody here also with letters after their last name knows that troubleshooting for 9 months is not exactly the most fun thing in the world. So you feel you're just breathing heavily, wondering, why is this not working? And then there's a brilliant solution. For anybody interested in sucrose preference, I feel almost contractually obligated to unveil the brilliant solution. Thanks to two absolutely brilliant undergraduates that I work with in the lab who have saved my life on many occasions, the intervention that we needed to get our animals to actually lick was to put a glove box. So we put the glove box on its side. The animals have a couple of centimeters of a corridor. It's like 100% lick rate. They all find the lick spouts. They all lick it. And then when that happens, now these guys here in the orange, again, the positive memory group, you see this rescue in sucrose preference back to the levels of control on stressed animals. So interestingly, and this is more fodder for discussion afterwards, just wanted to show that when we measure anxiety-related behaviors, as measured in the open field or in the elevated plus maze, the orange line here, again, is our positive memory group. We don't see any rescue in those two tasks. It could be for multiple reasons that we could discuss afterwards, but just to put it out there, intervention seems to be specific to tests like the tail suspension test and the sucrose preference test, and we haven't gotten any effect in any anxiety-related measures. So one of the important questions that we had in mind now was, well, what is a positive memory doing? If we reactivate it, we've sort of been treating the rest of the brain as a black box. What else lights up? We know that in mood disorders, for example, there's a lot of reward circuitry that's impaired. So we wanted to know, when we turn these cells on, what else downstream is lighting up? What's potentially mediating these antidepressant-like effects? So we have a host of brain regions that we can go in and look and ask, what's turning on when we turn on this positive memory? We go and we measure, again, the activity-dependent gene CFOS. We just measure CFOS protein levels as a marker, an indirect marker of recent neural activity. So we have a group in which we stimulate a positive memory or a neutral memory as a control. So, okay, we see lots of increases in the basolateral amygdala, in the nucleus accumbens, shell, not the core, the lateral septum, and various subregions of the hypothalamus. So that gives us some hints as to where this positive memory might be traveling throughout the brain. We don't see any changes, also for many reasons that, it might just be the marker that we're using or so on. Globally, we don't see any changes in the MPFC, the habenula, the ventral hippocampus, which is interesting. And then in the VTA, it's P of 0.07, so I guess it depends if you want to squint or not, if it's statistically significant. I lumped it over in the non-significant part, but there could be something there. So for a variety of reasons, we wanted to focus on this loop between the amygdala, accumbens, and medial prefrontal cortex. Could we actually go and dissect the circuitry that could potentially be mediating the behavioral effects of reactivating this positive memory? So, okay. We know that in humans, for example, during the recall of particular kinds of positive memories, you see ventral striatal activity consistent with its role as part of a reward circuit. So we go in and we do a reasonably complicated experiment where we shine light into the hippocampus to reactivate this positive memory. And now we inject a virus in either the basolateral amygdala, again, an area that blew up with activity, or in the medial prefrontal cortex, an area that did not blow up with activity. Now, this virus, ARCH-T, just allows us to inhibit brain cells, right? Channel rhodopsin activate, ARCH-T inhibit. So what this means is we can inhibit in real time the terminals from the BLA to the accumbens. We're clamping down on those terminals while simultaneously reactivating the hippocampus. And the exact same logic for the MPFC. We're inhibiting the terminals from the MPFC to the accumbens, but while simultaneously reactivating the hippocampus. What this looks like is you need, in this case, 2 lasers, green and blue, a mouse that has 4 implants, 2 over the accumbens, 2 over the hippocampus, and then you need to connect them to a lot of laser to try to now modulate this behavior in real time. So what happens when we do that? This is ARCH-T in the BLA, and this is expression of that virus to inhibit This is expression of that virus in the terminals so that we can inhibit the terminals in the accumbens, similarly for the medial prefrontal cortex to the accumbens as well. So the way we go about this behavior is we start off with all of the lights off. So the animals are all struggling at a baseline level. The orange group is the group, the important one, where we inhibit the BLA to accumbens terminals. So lights off, all animals struggle at similar levels. Now, when we turn the green light on, in this case, to inhibit the terminals, and the blue light on in the hippocampus to activate it, we see a blockade of our rescue. So in other words, when we turn the blue light on in the hippocampus and reactivate this positive memory, it seems to require the real-time activity of the BLA's terminals onto the accumbens. Because when we release the green light, and we still keep the blue light on to activate the positive memory, now we see the rescue that we saw originally. And we can do this in the other direction, where we turn the blue light on first. All groups show an increase in struggling. We turn the green light on to clamp down on these amygdala terminals onto the accumbens, and we see a modest deficit now in struggling. So importantly, this translates over for the sucrose preference task as well. If we have the inactivation of the BLA terminals while activating the hippocampal-mediated positive memory, it seems to block the effects. Whereas when we release the green light and only shine blue light into the hippocampus to activate those cells, now all animals show this increase in struggling back to unstressed animals' levels. So we think that by reactivating these hippocampal cells, we're forcing these BLA terminals onto the accumbens to come online, and again, they seem necessary for the antidepressant-like intervention that we have via positive memory. So to end, one of the things, a very rightful criticism of optogenetics is usually, well, that's not really the way it works in people, right? You give one dose of Prozac, because we're not going to do much. You give weeks and weeks and weeks, and you start seeing some effects in some people. So we thought, what if we could just go in and reactivate these positive memories chronically? Like, what if we could just do it twice a day for multiple days over and over and over again? Can we induce changes in the brain now long term? So in other words, all of the experiments before, we had to shoot light into the brain to get our behavioral effects. But when we turn the light off, the animals go back to that depressed-like state. So what if we could make a kind of intervention that outlasts real-time light treatment? So for these experiments, we either label a positive memory or a neutral memory. We put the animals again through this chronic stress protocol, and then we either don't reactivate anything as a control, we reactivate a positive memory once a day, or we reactivate a positive memory twice a day for 5 days. And now what we see is in the tail suspension test and in the sucrose preference test, compared to the group that had 1 day of stimulation, no stimulation, or neutral memory stimulation, or no stress, you see that the group that had 5 days of stimulation both struggles more and prefers sucrose more than the other control animals, and it's not statistically significant than animals that weren't stressed to begin with. So it seems to elicit a long-lasting rescue of these particular behaviors. So lastly, we thought, okay, so if we can reactivate these positive memories multiple times over and over again, and this seems to actually correct some of this behavior, can we find any neuronal correlates that actually can begin to correlate with the rescue of this behavior? So this led to months of a giant witch hunt, and I lost actually a bet and a really good bottle of gin to this because it was the one thing that I didn't think was going to work because it was too clean and too neat. But these are adult-born brain cells in the dentate gyrus of the hippocampus. And the strongest correlate that we found in these animals that were rescued was an increase of neurogenesis. So animals that are not that haven't undergone this stress protocol show a lot of, these are adult-born cells, they show a lot of neurogenesis. Animals that were not stimulated, again, they did not have a positive memory stimulated, but did undergo the stress protocol showed decreases in neurogenesis. Stimulation, importantly, of a neutral memory twice a day for 5 days did not seem to reverse the effects of stress on neurogenesis, whereas 5 days of positive memory stimulation actually robustly increased, in this case, neurogenesis, or reverted the neurogenesis levels of stressed animals back to unstressed control levels. So How this is causally contributing to behavior, we don't know, but it's a neat correlate that, I look at it as you're forcing these positive memories to become reactivated over and over again, and the brain begins fixing itself perhaps by making new brain cells, right? It's an outstanding hypothesis yet to be tested. So, the stuff that we've learned so far involves, the proof of principle that we can use this quote-unquote memory manipulation technology, we can bend that arc now of memory research, we can bend it towards therapeutic value, or potentially even translational value, you can imagine. So we've seen that we can acutely reactivate these dentate gyrus cells that we think are holding on to part of a positive memory, and we can reverse the effects of stress in these, as measured in the tail suspension test, sucrose preference test, again, traditionally used for depression-like behaviors. We know that they require real-time activity from the basolateral amygdala to the accumbens. There's certainly more going on, and I think that we just found a crucial node that this positive memory has to go through in order to translate to motor output. And then finally, we can reactivate these memories multiple times to mimic chronic stimulation protocols, and this can elicit long-lasting rescues in behaviors, which interestingly correlates with neurogenesis. So one of the things, or a handful of things that I'm interested in my lab in the future is really now bridging this idea of memory manipulation, bridging these techniques with psychiatric disorders, in rodents and across the evolutionary ladder to the extent that we can actually provide these interventions. So for example, all of the interventions that I talked about today were post-stress. They were when these maladaptive behaviors were already precipitated. Well, what if you just flip that manipulation around? What if you chronically stimulate positive memories prior to stress? Can you induce a kind of psychological protective net over the effects of stress in the brain? And there's interesting human literature around this so-called optimism bias and so on. Or alternatively, could you artificially reactivate negative memories over and over and over again, and could that make the animals more susceptible to the effects of stress? Or could that even begin to mimic some of these symptoms seen in mood disorders? We can also ask, well, why was there no change in anxiety-like behavior? There's good reason to believe that the anxiety-related behaviors might be mediated more through ventral hippocampal communications with areas like the basolateral amygdala and the bidirectional terminals there that are known to be heavily involved in anxiety-related behaviors. Or another one, what if we go in and ablate neurogenesis? Does that now prevent these rescuing effects of reactivating a positive memory? Interesting question. And finally, if we really want to try to combine sort of translational and back-translational approaches, well, what if we can find the brain cells that are holding on to a particular component of a memory throughout different brain regions, the MPFC, the amygdala, the accumbens, the works, and what if those specific cells that were active during a positive experience, pre and post manipulation, can we pull them down, look at their transcriptional landscape, look at all the hits, and now really start building this bridge across the evolutionary ladder by having these new, very amazing techniques in transcriptomics and so on. So I think we can actually have a rationale behind these kinds of interventions given the hits that we might find. Okay, so I have no idea how I'm doing on time, but all that said, it's really been, I guess right now I'm in that kind of waxing poetic mode because it's like bittersweet to leave grad school. Everything in lab has been a complete, honestly, a team-oriented effort. Like, it's something that, it's been 5 or 6 of us working on any given project. There's a real synergy that happens when people actually collaborate and work together, and it's one of those no-duh things that when people work together, more happens, but I've been really privileged to work with an absolutely remarkable team in the lab where everybody does a lot of everything. And it's amazing. Grad school is something that I can thankfully say has never really jaded me. If anything, it's made me more excited about the real revolutions that are happening in neuroscience right now, which I would argue is really just finally hitting its stride. And it's a nifty job to be able to say, your job is to make the unknown known. That's exactly what science is. It's a privilege to be a part of it and with the remarkable supporting cast in the lab. So with that, thank you.